When Kyle and Nicole welcomed their baby boy, KJ, they joked he looked like a giant peanut. But that newborn joy lasted exactly one day. Just 24 hours later, KJ collapsed into severe lethargy, surrounded by a maze of intensive care tubing. Doctors quickly diagnosed a 1 in a million condition called CPS1 deficiency. It's a genetic anomaly where the infant's liver cannot process standard dietary protein. Instead of digesting it normally, his tiny body was converting that protein directly into toxic ammonia gas, and that ammonia was accumulating fast. It was traveling straight to the baby's brain. If the levels weren't brought down immediately, KJ would not survive. For a rare genetic disease like this, developing a new drug, testing it, and securing regulatory approval normally takes a decade. KJ didn't have 10 years. He barely had weeks. The traditional medical pipeline is built for mass safety and scale, not speed. For an infant with a fast-acting weak genetic error, waiting for the standard system to run its course guaranteed he would die in the ICU. Out of conventional options, KJ's medical team made a desperate move. They transferred him from his local ward to the Children's Hospital of Philadelphia to temporarily stabilize his ammonia levels. That specific hospital housed a laboratory run by Dr. Karen Musunuru. He is a prominent cardiologist who also operates at the leading edge of gene editing science. Dr. Musunuru looked at the ticking clock and proposed a radical alternative, bypassing standard drug pipelines to rewrite the infant's DNA at the source. The parents and doctors faced a stark choice: accept a 10-year waiting period they didn't have, or attempt an unproven, highly experimental genetic rewrite on a severely ill infant. Think of the human genome as a massive instruction manual of roughly 3 billion chemical letters. Out of those 3 billion, KJ's life-threatening disease was caused by exactly one incorrect letter. A single inherited typographical error was causing his liver to fail. Traditional CRISPR acts like molecular scissors to cut DNA strands, risking collateral damage. Instead, Dr. Musunuru used base editing. Like a chemical pencil eraser, it precisely locates the wrong letter, wipes it away, and writes the correct one, leaving the surrounding DNA completely intact. Shifting the mechanical approach from slicing DNA to gently erasing and rewriting a single character severely reduced physical trauma. That subtle difference made live patient gene editing highly precise and highly survivable. Converting that theory into a physical treatment required a massive logistical effort. Over 6 months, his team coordinated with hundreds of medical and pharmaceutical specialists to synthesize the custom drug and secure emergency government authorization. This genetic sequencing chart shows exactly what the lab was analyzing to pinpoint the specific sequence where the edit needed to happen. Even with the drug ready, the 3-hour infusion procedure was terrifying. There was a very real fear that altering one specific base pair among billions could trigger a catastrophic biological chain reaction in the baby's fragile body. But 3 hours later, the custom base editing drug took effect. KJ's liver processed dietary protein. The toxic ammonia stopped building up. After spending 307 days fighting for his life in the intensive care unit, baby KJ finally went home, playing and growing like any healthy toddler. Executing a successful genetic fix in just 6 months proved that customized treatments can bypass the traditional 10-year timeline without sacrificing patient safety. The undeniable success of this rapid medical intervention forced federal regulatory bodies to entirely reconsider their handling of rare genetic diseases. In direct response to KJ's case, the government established the Plausible Mechanism Pathway. It's a brand new legal framework explicitly designed to cut through bureaucratic hurdles and fast-track the approval of custom genetic medicines for terminal patients. The government followed up this policy change with serious capital, releasing roughly $150 million in public funding, specifically dedicated to researching rare genetic anomalies. KJ's recovery provided the real-world evidence needed to dismantle decades of rigid protocol, clearing a much faster legal path for millions of future patients. Dr. Musunuru's breakthrough earned him a spot on the Time 100 health list, and he has since established his own company, Verve Therapeutics. He is already scaling this technology from rare diseases to incredibly common ones. His next objective is to use base editing to permanently lower cholesterol, effectively creating a one-time genetic vaccine against heart attacks. By correcting a single misplaced letter in an infant's genetic code, Dr. Musoniru's team established a roadmap to address humanity's most common fatal diseases.